Maria Kinsey.
And my name is Elaine Liu.
In the past 4 weeks, we have been working through summer ventures at UNCW, in which we utilize machine learning models to predict the presence of hereditary cataracts and pathogenicity from alpha-B crystalline mutations. We'd also like to acknowledge our amazing instructors and contributions such as Dr. Chen, Dr. Yishi Wang, Dr. Ying Wang, Ms. Hall, Ms. Gray, Mr. Suggs, and Ms. Smith, and additionally the Summer Ventures UNCW Biotech class.
So this presentation is going to be split into 6 parts. First, we're going to give a biological background in hereditary cataracts. Then we're going to explain the data we received, and then the relationships we found in this data. Then we're going to talk about the models we used and how it fared on our data. And then we're going to talk about our findings and our future work. So there's a few problems with hereditary cataracts. First, it affects 20,000 to 40,000 children each year. And the thing is, the sooner hereditary cataracts can be identified, the sooner the proper correction can be conducted. If these are conducted, then children can have long-term vision problems and even blindness. However, the current process of identifying hereditary cataracts is quite tedious and can take multiple weeks. Another problem is the specific reason why gene mutations cause hereditary cataracts is currently unknown.
First, we'd like to go into a little bit of background on the biology side of cataracts. So, the eye lens is transparent biological tissue that focuses light into the back of the eye or the retina. So, eye lenses, otherwise known as crystalline lenses, have a very high transparency, but cataracts can form typically due to old age or to damage to the protein in the center of the eye lens. And this can cause partial or complete blindness due to the decrease of transparency of the eye lens that it causes. And hereditary cataracts are a rare condition of cataracts, they're a rare form of cataracts, that can cause blindness in younger individuals due to gene mutation instead of natural causes for like old age or damage to the proteins themselves.
So the main focal point of this research project is the alpha crystalline protein, which is a subunit or subprotein of the overall crystalline protein. They're most common, especially in the eye lens, and they're made up of 2 types of protein structures, as you can see in the image below. The repeated lines back and forth are called beta sheets, and then some twists and curls are called alpha helices. And so the reason why alpha crystalline proteins are important is because they're able to prevent the development of cataracts in 2 key ways. First, they protect against proteins from aggregating or coalescing as a result of heat, and second, they're able to refold denatured or broken down enzymes and proteins. So when you have mutations to the alpha crystalline, they can sometimes reduce this ability to actually protect and prevent cataracts from developing.
So with that being said, we begin asking ourselves if a machine learning model can be created to predict whether certain alpha-B crystalline mutations will lead to hereditary cataracts and be pathogenic. We also needed to gather more information about the role of certain biochemical attributes of mutations in predicting and causing hereditary cataracts.
Now, the original dataset that we received was from the biotechnology group's teacher's assistant, which consisted of 40 alpha crystalline mutation observations, 14 of which were positive and 26 of which were negative. And they also included several variables that we could use as covariates in our function in order to predict whether or not it was positive or negative. Now, we're going to be using the terms positive and negative a lot throughout the future of this presentation, and so we'd want to explain what they mean. So the positive group are observations of mutations that are known to be pathogenic and cause diseases, including hereditary cataracts, while the negative group are observations of natural mutations that are assumed to be benign and do not cause diseases like cataracts.
Here's a sample of the data we received. As you can see, there's both qualitative and quantitative data. Here are a few vocabulary words that we are going to be discussing throughout the rest of our presentation. First is isoelectric point, and it is the pH value which amino acids carry no net electrical charge. Solvent area represents the measure how much solvent the amino acid has access to. Delta delta G represents the change in Gibbs free energy, and a negative delta delta G represents the mutation destabilize the amino acid, and the opposite is true for positive. Evolutionary age is the amount of time the amino acid mutated from its original state, and p-del is just a standardized version of evolutionary age. Here are some characteristics of the amino acids we were provided. The most common were positive and negative charge, and there was also nonpolar and polar, and a specific amino acids also had characteristics.
So now moving away from our data set and towards looking at correlations and relationships between them, we mostly focused on numerical variables because we knew that those would be most important and most relevant for making models. Here are two 2D scatter plots of the many that we made comparing isoelectric point and p-del as well as solvent area. These are variables that at that time and throughout we thought had the most predictive power. And from what you can see here, isoelectric point specifically does a really good job of separating the data points into almost 3 separate categories, with the green or positive data observations above or below the middle red or negative observations, with the exception of like 2 outliers. So from the sort of exploratory data analysis that we performed, we were able to have a better idea of which variables we would use as covariates or sort of X variables in the models that we would make. We also wanted to compare not just 2 variables, but also 3, so we made some 3D scatter plots, again comparing isoelectric point, solvent area, and p-del values directly to see if there were any other outliers that we could see, as well as find any other ways that we could split the data ultimately with our model. And from what you can see, especially for isopoint, the best models we predicted would work non-linearly.
So as I previously mentioned, we were provided the characteristics of amino acids, and we were provided this data for before the mutation and after the mutation. So we decided to combine the characteristics into the before and after characteristic into one variable, and we found a clear correlation between the group and the isoelectric point, besides 2 outliers. As you can see, the negative group most commonly has an isoelectric point of 6.76, and the positive group has an isoelectric point both higher and lower than this value.
So once we thoroughly analyzed the data and found certain variables we wanted to test, we began using classification models to draw conclusions from the data. We began by resampling with random splitting, and this was repeated 40 times. And since we had a smaller data set, we set 75% aside for training, meaning this is what the model was molded around, and then the other 25% was the testing split or the testing group, so we could see the accuracy of these models.
Now, we tested out several different classifiers in order to figure out which would be best to use in our final machine learning model, and we're going to show you the best performing of these classifiers. Now, the first one is ADA-boosting, or adaptive boosting decision tree model. Now, it's a decision tree model, which means it works by dividing the prediction space into smaller and smaller areas. This can be pictured as a tree with branches growing outwards, which is why it's called a decision tree model. Now, it's adaptive boosting, meaning it works by learning from its mistakes. So it will try to classify the data, fit the data to the classifier, and then it will weigh the incorrect guesses or the incorrect predictions and adjust for its mistakes so that the next time it goes, it'll be less likely to make the same mistakes as last time. And as you can see here, it had around an average accuracy of 96% and an F1 score of 97%, using p-del and isopoint. So we can really see that those are some strong predictors.
The next best performing model, in no particular order, was KNN models, or k-nearest neighbor. These models work by classifying data points as the same class as what the majority of its nearest neighbors class would be. So the number of nearest neighbors that are compared to depends on a chosen k, and for us, after running through multiple k values, we found that a k of 3 worked best. So looking at the 3 nearest neighbors to classify any one data point is how this KNN model worked. You can also see that it performed pretty well with a best average accuracy of around 95.5%, pretty high specificities or true negative rates, as well as very low runtimes, making it overall a pretty efficient model. You can also start to see isoelectric point be a consistently top-ranking variable as covariates, signifying that these variables would ultimately be really important in the final model that we chose. The final best performing model was SVC models, or support vector classifiers. These work by creating hyperplanes or border delineations that separate data points into different classes by being the farthest away from data points on either side. This one also performed very well, even better than KNN, with an average accuracy of 96.5 as the best, but consistently around 95, even higher specificities and precisions, and even lower runtimes, making it overall a more efficient model.
So again, those were the top best performing models, but we also tested several others. So this is a color-coded bar graph that displays that. And then this also compares the average accuracy, specificity, precision, and F1 score. And as the other previous models have shown, isoelectric point and p-del has been a strong correlating relationship between two variables that perform very well together. So another great opportunity we got in this class was getting to actually getting to collaborate with the biotechnology class, in which they gave us some data that we got to add to ours to make it not only more give a bit better variety, but so we could also have a more accurate testing model.
So we received 4 new data sets around the same size as the original one from each of the biotechnology groups. And while they varied a bit, we could still see that KNN, SVC, and adaptive boosting remained in like the top 4 models for each of the groups. And they were consistently the most high among all 4. Here you can see group 1 and group 2. And then here's group 3 and group 4. And you can really see that KNN, SVC, and ADA boosting maintain around the top 3 for all of the models tested.
After taking in various criteria, we decided to choose a support vector classifier as our final model. This is because it had a shorter runtime compared to ADA boosting, and also was less susceptible to future outliers compared to the KNN model. Our final model had an accuracy of 95.3%, a sensitivity of 90%, and a specificity of 98.4%.
Now, using the support vector classifier with isoelectric point and p-del as covariates, we were able to make a prediction tool that would predict the likelihood of a mutation being pathogenic when we give it its biochemical characteristics. And so basically what that means is we'd be able to input a data set like the ones we received and be outputted a table like this one, which has the index of the specific mutation and its confidence value for either positive or negative, and whichever confidence value is higher, it would decide as its final prediction of whether or not it believes that this mutation is positive or negative. Now we wanted to make this tool as user-friendly as possible, so that researchers who do not have a background in machine learning would still be able to utilize this tool, as well as medical professionals trying to predict the likelihood of someone having hereditary cataracts. And so we decided to make it an executable file in which you'd put a dataset CSV into an input folder and run the exe file and receive an output CSV file which had what was shown on the table before, which would be the index, the predictor, and the confidence values for either positive or negative.
Ultimately, there are 2 key findings from our research project. First was that non-linear models, or KNN, SVC, and ADA boosting specifically, were really good at predicting pathogenicity of alpha-beta crystalline mutations, with, of course, SVC being the most efficient overall. And second were variables, or specific variables like isoelectric point and evolutionary age as p-del, that actually were able to give more explanation biologically as to why certain mutations over others led to hereditary cataracts and add more to the literature space surrounding hereditary cataracts. There were also 2 key contributions. First was a novel method and approach of predicting pathogenicity and presence of these hereditary cataracts from mutations with high accuracy of 95.3%, but also in a much shorter amount of time plugging and chugging certain CSV files. But we were also able to create this predictor function that Sam mentioned that makes it more accessible to actually detect these mutations in a more medical professional space.
There are also many other steps that can be taken to further improve the accuracy and use of machine learning models in predicting the pathogenicity of specific mutations. For example, we would like to work further with the biotechnology students to gather more training and testing data to improve the model as time passes. We would also like to continue testing hyperparameters and implement cross-validation to ensure the most optimal model is selected. And then lastly, we would also like to be able to develop and use this function so it's easily accessible in medical spaces for children who need this treatment more urgently. These are our references. And thank you guys for all watching our presentation. Do you have any questions?